Thank you so much, Randy, for that introduction. And I also did not trip coming up here, so that was a plus for today. Okay. So as Randy mentioned, I'm an associate professor here at Columbia. I've been here almost 20 years, starting as a grad student. And I'm going to be telling you a little bit about our work in the lab to develop drugs that you could administer before stress to protect against stress. So most times that you are stressed, right? We've all been stressed in this room. You have a severe stressor, and this in some patients will lead to PTSD, depression, anxiety. We heard from Randy, suicidal ideation. And what's typically done is then you treat the patient with psychotherapy, antidepressants, a number of different treatments, but we wait for the patient to exhibit those symptoms. What if we started to think, can we bias this population that is susceptible to a resilient population? Why are all individuals undergoing stress that will not develop depression or PTSD? Can we make them resilient to the stressor? So that's kind of the broad question I'm going to be talking about today. And we are taking a clue from most fields of medicine where we use the term prophylaxis or protective measures that you can give before. Some examples that I'll show here. I'm assuming most of you have heard about the Truvada on TV. It's a commercial, it's on all the time. If you get exposed to HIV, they're often given Truvada to prevent the development of AIDS. If you go in for surgery, you often get antibiotics before you go into surgery to prevent infections. I have 3 small young children at home. They've all gotten the Hep B vaccine, trying to prevent hepatitis. If you have high cholesterol, right, or you want to, prevent heart disease, you take statins. If you're getting dental surgery, you take antibiotics. The list goes on and on. We can have, methods as well as taking a pill or a vaccine where you can exercise to prevent age-related cognitive decline. You can exercise to prevent heart disease. We haven't really taken that thought yet in psychiatry. You heard a little bit today about Randy using the smartphone, but what if we could prevent these things from happening before they occur? So what my lab has been trying to do, and I will preface this with, we did not set out to do this. It was a serendipitous finding that has changed the course of the history of my lab. We give a single injection of a drug 1 week before stress, and that allows for long-lasting protection so that if you are exposed to stress, you have what we'll call the resilient and not susceptible phenotype. This is a great way to protect against cases in which a stressor will occur. So you can think about forward deploying troops in the military, humanitarian aid workers, firefighters, police dogs. I mean, the list goes on. There are certain cases that you could predict a stress from occurring. However, that's not the case for most people, right? I knew Randy was going to invite me to come give a talk today. I'm prepared, but we don't always know when the stressor will be. So there's a secondary time point that I want to discuss, which is giving a drug immediately after the stressor, not when the patient develops PTSD. Could we prevent at that time point as well? And what I'll show you today are 2 data sets showing that it is actually possible. So I'm the only one up here that's using rodent models, but we use rodent models to understand the neurobiology and mechanisms that are underlying this disorder. And while I think mice are great, they are in a controlled environment, they're mostly genetically identical. We can't ask a mouse, are you stressed? Are you depressed? Are you sad? Do you have suicidal ideation? So the field has had to develop techniques to stress the animals as well as measure their behavior to see if they are exhibiting a depressive-like phenotype or an anxiety-like phenotype. So I'm not going to go through all of the models and the tests today, but I'll broadly just talk about some of the main models that we could use. we heard just about an early life stress. Most people will realize that early life stress is so different from an adult or a later life stress. And so we could model early life stress in rodents as well as later life stress. We could use fear-based stressors. There's all different types of stressors we can use, as well as we can use biological stressors, so neuroendocrine models where there's not a physical stressor as well. So these are the tools to stress. But then we have to ask the mouse if they are depressed. And there are a number of assays that we could use to get at these phenotypes that we're interested in. So you can measure apathy, anhedonia, anxiety, social behavior, despair, fear generalization, fear memory, or perservative. These are just some of the traits that we can assess in mice. And then using a compilation of the various stressors with the various tests, figure out if the mouse is a little depressed or if your drugs are working, and vice versa. So the first study, as I mentioned earlier, was, I swear, not what we were trying to do. We were anesthetizing mice with a drug called ketamine. Dr. Mann will discuss this later. Ketamine has revolutionized the field of psychiatry. You probably also know it as Special K. It's on the World Health Organization's list of most essential medicines. It is an anesthetic, and we were using it as an anesthetic in mice to anesthetize them for a totally different set of experiments. And we kept finding really long-lasting effects of the ketamine. And through many, many different experiments and convincing my PI at the time that this was interesting, we found that a single injection of ketamine 1 week before stress could basically protect the animals completely against that stressor. So I'm just going to show you some of the work that was done today by 2 grad students that were in my lab. And we really have expanded this finding since the initial finding. In the first set of data that we found, we gave 1 injection of ketamine, again sub-anesthetic, 1 week before a chronic stressor. And we looked at depression weeks later and found that the animals that got the single injection of ketamine a week before were back down to control levels and not as stressed at all as the stressed animals. We also used a PTSD-like model, and in this model, the animals got a single injection of ketamine 1 week before the PTSD model, and you can see that ketamine attenuated this behavior throughout the testing period. We also used a fear-based assay and found that ketamine blunted their fear response. They never mounted the same fear as the control animals. They extinguished their fear as the control animals are the same, but they didn't mount the same fear response. So I'm not gonna, for time limits, we verified this in a number of different stress models, dosing, you name it, sexes. But it was a really robust finding that we were, just kind of blew our minds that no one had found this before. And so I mentioned earlier, I work with mice, and I thought, okay, no one's really going to pay attention to this. It's in mice, et cetera. And we started getting a lot of press from the initial paper. And lo and behold, since that finding, a number of studies have come out verifying our work in humans. And so I want to focus your attention, which I'm behind here so I can't see, on the paper that's the correlation between ketamine and PTSD. This paper had come out, and we were unaware of it initially, but they looked in Iraqi war veterans that were burned on the battlefield. And those veterans that got ketamine on the battlefield to deal with the burn, they had hypothesized they would have an increased prevalence of PTSD. But actually, the soldiers that got ketamine had a 50% less prevalence of PTSD than those that did not get the ketamine. Since then, a number of studies have verified prophylactic ketamine in women with a history of recurrent postpartum depression. And in these studies, the women have given birth, and then they get an infusion of ketamine right after birth, and it's significantly decreased the incidence of postpartum depression. And a more recent study by Dennis Charney and James Murrow has come out verifying that prophylactic ketamine can work in healthy adults that are stressed. So, we use mice because they're models, but I'm realistic that we would like these findings to get out of my lab and help the people that need it so much. And so the fact that, this paper was published less than 10 years ago, and it's now been verified in a number of different rodent models and has actually gotten out into the human population is what we want to do with my work. So the next couple slides, I'm going to show you what we've done since then that's trying to move this forward and not just stay on ketamine. Although I do really love to talk about ketamine, but we're going to move on. So is ketamine the best drug to act as a prophylactic? And I'm assuming if many of you have paid attention to the news recently, you know it has been in the news a lot. Do I think ketamine is the best drug? No, we don't. We are trying many other things to figure out what is the mechanisms of action, and if we can figure that out, can we make better drugs or understand different drugs? So we have collaborators. Brianna Chen is one of my grad students, now postdoc, and a number of collaborators at Paris/K. And we thought, let's try other drugs that don't act like ketamine, and we thought they would be a negative control. And so we used drugs that were targeting the serotonergic system, and one of the drugs we used is called prucalopride. It's an FDA-approved drug for chronic constipation. If you're interested, it targets the 5-HT4 receptor. And we thought, okay, this is not going to work. Like, let's just try it. And lo and behold, we did the same thing where we give them a single injection of ketamine or one of these 5-HT4 agonists or the serotonergic drugs 1 week before stress. And we found that the animals that got ketamine, they had reduced fear and reduced depression, like I showed earlier. But the animals that got prucalopride at a very low dose, one injection of 3 mg per kg, had reduced fear and reduced depression. We verified these effects with another drug that targets the serotonergic system. It's got a bunch of letters and numbers. I'm not going to ask you to remember that. We'll just call it PF for now. And that drug also worked at a very low dose at 10 mg per kg, one injection, 1 week before the stressor. And I want to mention this with all of the drugs we're testing. The half-lives are very short. They're out of the system in a few hours. So they're nowhere near on board when the animal's stressed or anything. So it is amazing, right, that a single injection of a drug that shouldn't have effects, right? Many of us have got anesthetics. We think, okay, it's one time, has such long-lasting effects, and now you're moving it outside of the typical canonical target for ketamine to other targets that we're thinking about. So this is great. We thought, okay, we've moved past ketamine into some serotonergic compounds, but what can we do about developing our own compounds? And so we've been working in collaboration with, his name is Gilles Rubenstein, and again, my postdoc, Brianna Chen, to figure out if we could figure out more drugs that are working, what is the common mechanism between them? And here we've been developing a drug we'll called FENM. FENM is an analog of memantine, which is what I'm assuming most of you have heard. We've given it a single injection 1 week before stress on the same assays. And what you can see here is that ketamine in blue and FENM in red at 2 of the doses decreases fear and decreases depression following the stressor like ketamine. What's really interesting is that the parent compound, memantine, shown in the black bar, does not do that at all. So it's not specific to that pathway. It is a pretty new and novel molecule that we're hoping will be great. So I mentioned earlier that there are cases that are great to know that that person's going to get stressed, but I would say 95% of us don't know we're going to be stressed. And so we really wanted to expand the time window from this 1 week time window to see if these drugs could work after stress. So in this set of experiments, we gave ketamine 5 minutes after the stressor, not 1 week before, and assayed fear and depression. And we also gave the new drug that I had mentioned, FENM. And what you can see on the left is that none of these drugs work when given after against fear. If you remember earlier, their fear would always decrease. But if you look on the right, the animals that got an injection 5 minutes after the stressor, they have decreased depression, whether they were given ketamine or any of the doses of FENM. And so we thought this was really promising because now we are not limited to trying to predict who will be stressed, but that if you did have a stressor, you could potentially decrease the depression, but not the fear, giving it after. So everything I've shown you today, and I didn't mention it on purpose, was in male mice. We've done a lot of female mice as well, but it's realistic. I'm a woman that females get stressed as well. And so I want to also add this point here that if we gave an injection after the stressor to female mice, we also see that decrease with FENM beyond what can be done with ketamine. And so we really think this is a novel compound to develop because it is efficacious in both stresses. So the last data I'll show you for today is something that's totally on the forefront, not fully published yet. And we took a lead from the cancer field where, one drug is not a one-size-fits-all, right? You might need a combinatorial treatment or a suite of compounds that could work. And I focused today, for simplicity, on depression and stress, but there were all those other traits that I brought up that we haven't really discussed: anxiety, feeding behavior, locomotion, social behavior, although ketamine does work really well for social behavior. So we thought, could we combine ketamine and the prochloropride, and what would happen if we did this? And so we gave a single injection of either drug or combined at a varying number of doses, 1 week again before the stressor. And I'm just going to show you the behavior we obtained with fear, depression, and what's called hyponeophagia, which is basically eating in an anxious environment. So there's a lot of bar graphs and a lot of numbers. I'll just broadly tell you what we found. 1 dose, again, each works for ketamine, and 1 dose works for prochloropride for the fear. And when you combine it, you can see that you can lower the dose of the ketamine to get it to work. In the depression, we get 1 dose each to work again, and then when you mix and match the combinations at different doses, you can again lower the ketamine and get it to work. And so we thought, okay, this is okay. They work separately, they work together. But when we looked at hyponeophagia, what we found is that only when the drugs were combined together did they actually decrease this behavior. Now, this is amazing to us because we had never seen an effect in this test earlier. You're basically assaying the animal's anxiety to go eat something in a brightly lit environment. And so we're hoping to start to think about, everything we have focused on is stress-induced depression and fear. We could potentially apply this to eating disorders and see if it's going to change behavior, but maybe you'll need the combinatorial drugs and not either drug alone. So what's next? I hope I didn't overrun you with data, and I just want to summarize what I've shown you today. It's changed my lab, it's changed my life because we haven't set out to make prophylactic compounds, but the effect is so robust that it was just hard to walk away from as a scientist. And so I've shown you that a single injection of ketamine 1 week before stress is protective against stress. We don't have to wait for the animals to get full-blown PTSD or stress-induced depression and then try to figure out a regimen we could potentially protect before it starts. I then moved on and showed you that we've had a number of other compounds identified. I've only discussed, 2 or 3 others today. We have a suite back in the lab, and we're trying to get these compounds out of the lab into development so that someone can pick them up and run with them and, ideally apply them to not just the stress-induced psychiatric disease that I had discussed today. And the last part I want to mention, which I did not discuss today, is that, my lab has a whole other part, and we study learning and memory, like Randy had mentioned earlier. And what we specialize in is identifying and manipulating neural ensembles across the brain. And we have this technique to visualize brains with single cell resolution in situ. And so what we've been doing with a lot of these compounds and stress models is to be giving the drugs with the mice and looking at the brains, across the brains. Can we figure out the individual ensembles that are mediating this behavior? Can we figure out the ensembles that are actually rescuing this behavior when you give this drug? What are the circuits? What are the nodes that are doing that? And so I've been really thankful for now an assistant professor, Alessia Mastrodonato, for leading this in my lab and really helping us figure out, if we can figure out where these drugs are working, can we make even better drugs that are targeted to kind of fix the ensembles and fix the behavior? So I hope I gave you some hope today, and thank you so much for listening, and that's it. Thank you, Randy and John.